Great. So, hello, everyone. I'm Maura de Koster. I'm a PhD candidate at the Vrije Universiteit of Amsterdam, where I use isotopes to look at the mobility and diet of the people living in on the lower Germanic lemurs. My PhD is part of the Constructing the Lemurs projects, which looks at that same lower Germanic lemurs from a variety of perspectives, with an emphasis on how borders function. Today, I will present preliminary isotope data from the human remains from the cemeteries at Valkenburg, which is the most western site on the European mainland. As you can see here, Valkenburg is situated right on the coast along the lower Germanic lemurs, which is red line. It was one of the earliest legionary fortresses in the region of in this region of the Roman Empire, and one of the most popular assumptions is that it was built to accommodate the troops that would then go on to invade the British Isles. However, what makes it a unique place to study the human remains is that it contains the biggest cemetery west of Nijmegen, which was, of course, a huge Roman city. The biggest cemetery in Valkenburg contains about 700 graves. This is by far the only cemetery of this size that we find west of Nijmegen. Between Nijmegen and Valkenburg, we only find some smaller cemeteries of less than 100 individuals. Of these 700 Valkenburg graves, around 145 graves were inhumations. This is again an unusual high number of inhumations for the region in the Roman period. The majority of the inhumations were infants, but 35 individuals were adults. The size of the population buried in Valkenburg provides a unique opportunity to study border dynamics along the lower Germanic lemurs. So the Roman military arrived in Valkenburg around 40 CE and installed a large legionary fortress, so presumably to base those troops that were meant to invade the British Isles. The fortress existed for a maximum of 10 years, so not that long, after which it was demolished by the Romans themselves. After the demolition of the legionary fortress, the military presence was reduced to at least one, but probably a few more smaller forts. It is the period after the legionary fortress which is studied in this research. So after demolition of the legionary fortress, the biggest cemetery, Valkenburg Marktveld, appears between 50 and 70. This is a cemetery where where about 700 individuals were buried. However, in the first quarter of the 2nd century, two smaller cemeteries located very close to the big cemetery, Marktveld, appeared. In both of the cemeteries, Weertkamp and Vliegveld, around 15 cremated individuals were found. Those two smaller cemeteries were linked to smaller farmer settlements in Valkenburg. All of the cemeteries are in use until the late 3rd century. There is, however, evidence for people living in Valkenburg in the 4th century as well. So this was the timeline, but I would also like to introduce more of the geographical situation as well. So here we have the legionary fortress that was built in 40 and only existed for a maximum of 10 years. So that disappears. A smaller fort still exists. That's the red square north of the map. And then after the legionary fortress is demolished, the Marktveld, which is the orange on the map, appears. So this is the big one where about 700 individuals were found. And then about 100 years later, the two smaller cemeteries, Weertkamp, which is the green, appears, and then Vliegveld. It's not the entire red dotted line, but it's situated in there very close to the other cemeteries as well. So I'm going to go into the actually human remains, beginning with the inhumations. I've already explained how the high number of inhumations is unusual for the region, but the adult burials from Valkenburg Marktveld, very large cemetery, are interesting in other ways as well. The adults were all buried in a careless way. There were a few prone burials. Some of them seem to have just been thrown in the grave. Most of them didn't have any grave goods, and if they were, it was a singular pot like you see on the photo. Osteological analyses showed that these individuals performed heavy labor during their lifetime, and they were in general taller than the average population that was living in the Netherlands in the Roman period. So this raises a lot of questions. Why weren't they cremated, and why were they buried in such a seemingly careless way? So this is where the isotopes come in. For the inhumations, tooth enamel was sampled. If available, first, second, and third molars were sampled, so individual life histories could be constructed. The first molar represents the first 3 years of life, second molar the 3 to 8 years of life, and third molar 8 to 16 years of life. Strontium, oxygen, and carbon analysis were done on all of the enamel samples. Next to the inhumations from the big Marktveld cemetery, we also analyzed a few individuals from one of the smaller cemeteries, the Vliegveld one, where we sampled a long bone and a rib, which had to be fully calcined, which means burned until they were completely white. The long bone represents the strontium intake from the last 25 years of life. A rib has a higher turnover rate compared to a long bone, so represents about the last 5 years prior to health. So in total, 36 individuals were sampled, of which 28 were inhumations and 8 were cremations. Both the strontium isotopes as well as the stable isotopes were analyzed using standardized methods. The strontium isotopes were measured on a thermoionization mass spectrometer, and the oxygen and carbon were measured on a stable isotope ratio mass spectrometer. So let's start with the results from the carbon and oxygen from the inhumations. So these are the enamel samples. The triangles on top are the oxygen, while the dots on the bottom are the carbon. The pinkish bar represents the range of the oxygen and carbon isotopes we would expect from local individuals. So with the exception of 35 and 46, all individuals exhibit delta 13 carbon that are compatible with a local C3 diet. Based on the oxygen isotopes, 7 individuals deviate significantly from the expected regional oxygen isotopes. Intra-individual variations between the first, second, and third molar are observed as well. This next slide are the strontium isotopes from the inhumation still. So again, these are enamel samples. So we could already see some mobility from the oxygen and the carbon isotopes, but when looking at the strontium isotope ratios, the high degree of mobility among these individuals becomes really evident. Again, the pinkish bar is what we assume to be the local range. Strontium isotope data here shows both great inter and intra-individual variability, which we can see in the difference between the first, second, and third molar. Definite non-local origins and intra-individual mobility is evident in 57% of the sampled populations, except for child 36. All non-local strontium are higher than the expected regional strontium signature, which we expect to be between 0.7088 and 0.7095 based on the bioavailable strontium map from the Netherlands. So as I already mentioned, in addition to those 145 inhumations where we sampled 28 individuals, more than 700 cremation graves were also excavated in Valkenburg. However, to date, we only sampled the ones from Vliegveld, which were possible. And these are the long bone and ribs fragments of well-cremated individuals. In contrast to the majority of the strontium isotopes from the Valkenburg Marktveld inhumations, initial strontium data from the cremated individuals show a local origin with all the ratios between what we would expect from local origin. albeit, as you can see, on the lower side of the local range. As I said in the beginning, I'm presenting preliminary results from Valkenburg, but from these two data sets, a few careful conclusions can be drawn. For now, the high mobility in the inhumation is a strong contrast with the very homogeneous strontium ratios measured in the cremations from the smaller Vliegveld cemetery. This could relate to the different communities who lived in Valkenburg, but the data set needs to be expanded to include the cremations from the big Marktveld cemetery and the other smaller Weertkampen cemetery before definitive conclusions can be made. Another important thing to note is that the data sets from cremations and inhumations are not easily compared because we are looking at childhood mobility in inhumations, while cremations are giving information about the mobility from the last 25 years of life. Cremated bones also gather strontium from the time when the individual was already living at Valkenburg, which, depending on how long they stayed at Valkenburg, could make their signal appear more local. Both of these elements make it so that mobility will often be more obvious in inhumations than they are in cremations. So, like I said, this is preliminary, so we have lots of plans for this site as well. Lots to do. First priority is to include the strontium isotope ratios from the other two cemeteries. Those are being sampled right now, so we can compare the Marktveld inhumations to the Marktveld cremations, as well as compare the cemeteries from the two smaller farmer cemeteries to cremations from the big Marktveld cemetery. The inhumations from Marktveld will also be radiocarbon dated because the lack of grave goods made it impossible to date these graves other than it's after 50, to know if they were all buried together in a short period of time and to when they were buried in relation to other graves will possibly provide some new insights into what happened to these buried individuals. So, interestingly, 20 years ago, about 10 samples were taken from which lead concentrations were measured, which showed elevated lead in the individuals buried in Valkenburg. So the plan is to take new and more samples to really look into the role lead played in the Valkenburg site. We have also sampled for ADNA analysis to be taken so we can, from the inhumations, so we can gain some more insight into ethnicity and possible origin of the individuals buried that were seemingly carelessly buried at the Marktveld cemetery. We will also make a local updated strontium isoscape for the Valkenburg site and the region around it, so that all of this strontium data that we have measured at the site can be contextualized a little better. And finally, the dataset will be implemented into the bigger Constructing the Limes framework, where we will try and combine all of the different perspectives and insights to make sense of the Valkenburg site and in a more broad way, the Lower Germanic Limes in its entirety. So I've just put on some information about the project and my own personal information if you would like to see what we are doing. But thank you so much for listening, and I'm happy to answer any questions you might have.